Carbohydrate biochemistry is oh so sweet, and it goes way beyond that sugar that you eat. Speaking of which, that sugar that you eat is probably sucrose, which is an example of a disaccharide. It's made up of two sugar monosaccharides, individual building blocks that are joined together through a glycosidic bond. In the case of our sucrose, we have a molecule of glucose and a molecule of fructose that are linked up to give us sucrose. Now, that glucose, that's kind of our main monosaccharide. That's what we call our blood sugar. And it can be modified in a lot of different ways to give us lots of other types of monosaccharides. And it can be joined together with other monosaccharides to give us disaccharides, like our sucrose. And it can be joined up into even longer chains. We can get ones that are good for certifying cell walls, ones that are good for storing glucose for later use when you want some energy. We can attach sugar chains to proteins and to lipids, and we can attach lipids and proteins to sugar chains. And we can end up with a lot of complicated terminology. At its heart, though, what all these things have in common, what a sugar has in common, a carbohydrate, so we often use the word like sugar kind of loosely to refer to carbohydrates or carbs. And so what these have in common is that they have one water molecule per carbon molecule. And so you have this general formula. There's some modifications because, well, if you think about how do we add one molecule per carbon molecule, we're going to end up with molecules with a lot of OH, a lot of hydroxyl groups. And hydroxyl groups are going to be great nucleophiles. They're going to be great places for modifications. And so we'll get things like oxidation, amination, and amidation, and acetylation, lots of different linkages to different things. And so those OH groups serve as great linkage points. And we'll see lots of ways that we can link them and link things to them. I do not want you to get bogged down by a lot of the nomenclature that we're going to see, as well as by how we represent things. My goal for this lesson is for you to get these key concepts and to familiarize you with the way that sugar molecules are often depicted. Because there's different ways that we can show sugar molecules. We can show them in different representations. We can classify them a variety of different ways. And it can get a little overwhelming when you're trying to see whether things are the same or things are different. So I want to introduce you to this nomenclature because it's things that you're going to see in your textbook. It's things that you'll see out in the wild. And so I want you to be familiar with these and not tied up in them so that you can focus on these bigger concepts. And so we'll review these concepts, but basically I want you to know the difference between monosaccharides and disaccharides, polysaccharides, the different types of polysaccharides, and different types of glycoconjugates. Not necessarily the terminology, but the consequences. And so in order to kind of be on the same page, though, we need to start by understanding some of this terminology. We're going to build up through glycobiology, so sugar science, in the kind of ways that we can build up monosaccharides to form more complex things. So we'll start with those monosaccharides, those single sugar units. We'll go to disaccharides. We'll see how the sugar units can join together through what we call glycosidic bonds. We'll then see how we can join up longer chains to form structural polysaccharides and storage polysaccharides. Our structural polysaccharides, we'll see these long chains that are held together tightly, these long straight chains. And then for storage, we're going to have things with lots of branching. We'll then branch out and see how sugar chains can be attached to other things like lipids and proteins to give us glycolipids and glycoproteins, and how we can have peptides that are attached to glycans to give us peptidoglycans, and we can have peptides that are attached to sugars to give us proteoglycans. And so you might be like, what is the difference? Those sounds so similar. And the difference is that the main kind of constituent of when you have two things, the main constituent of it is going to be named second, and then the kind of modifying portion is first. So if we think about a glycoprotein, the main constituent is the protein, and then it's got a glycan or a sugar chain attached to it. Whereas with proteoglycans, here the sugar's the star, and we have lots of sugars that are attached to a long chainy protein to kind of hold things together. And then we can also have peptidoglycans, where instead of a long floppy protein holding our sugars in place, we have little peptides that are holding chains of sugar in place. And so you'll see that in like bacterial cell walls. And then we can have glycolipids, and so we could actually attach sugar chains to lipids. And so maybe when you have some lipids on your cell surface, and then you have some sugar modifications, and then those sugar modifications can help your immune system recognize your own cells and reject foreign cells. And so we'll see how blood grouping is actually dictated by glycans that can be presented on the surface of red blood cells through lipids and proteins and things. So lots of cool stuff that we're going to see with carbohydrates, but we're also going to see carbohydrates displayed in a bunch of different ways. And so we need to be able to recognize the different representations of carbohydrates. I want you to be able to kind of classify things based on their number of sugars and whether they're an aldose or a ketose, which we'll talk about. But you do not need to know the actual names of these aldoses and of these ketoses. You're not going to be made to go from Fisher projections to Haworth projections in this class, but I want you to be familiar with seeing them in both of those forms. So let's start with those monosaccharides and get some of this terminology cleared up. So a monosaccharide, mono, one, single, saccharide, sugar. So remember the word roots are really important. If you know those word roots, you can learn a lot of stuff. And so sac, we have sugar, we have glyc, that's also refers to sugar, and mono, that would be a single. We have a single sugar unit, a single sugar monomer. These monomers, however, might have different lengths of the carbon chains. And so note how many OH groups we have. Lots and lots of OH groups. And we have different lengths of our carbon chains. And we can have the OH group sticking out on different sides of the chain. This is what we call a Fisher projection, where the things that are going out to the sides are going out towards us, and the kind of straight chain is going backwards. So we'll get to that in a second, but for now, just focus on we have these chains of carbons with one water molecule per carbon. So you can see OHH, 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 OHH. And so lots of carbons, lots of oxygens, lots of hydrogens, and then we have different chain lengths and different positionings of a carbonyl. And so for most of these, what you're going to see is most of the carbons are going to have an OH or a hydroxyl group on one side and then just a hydrogen on the other side. And sometimes you might not see that hydrogen shown. But then there's going to be a place where you have a carbonyl group. And where you have that carbonyl group can be at the very end or the very top, which we typically draw them like towards the top. And we number starting with that carbonyl carbon, the carbon closest to the carbonyl carbon. So you can see that here our carbonyl carbon was at the top and we actually have an aldehyde. Whereas here the carbonyl carbon is going to be sandwiched between two different carbons. So we have a ketone here, so this is called a ketose. And we have an aldehyde here, so this is be an aldose. And so when you see that ending ose, think sugar. When you see that sac, think sugar. When you think glyc, think sugar. And those are going to be our sugars. And so we can classify them as aldoses if they have an aldehyde and ketoses if they have a ketone. We can also classify them by the number of sugars that they have. And so in this case, we have a triose. We have 1, 2, 3 carbons. Here we have a hexose. We have 1, 2, 3, 4, 5, 6 carbons. And here we have a pentose. We have 1, 2, 3, 4, 5 carbons. If we had 4 carbons, we'd have a tetrose. If we had 7, we'd have a heptose. So you can kind of see how the logic works. And again, that's where it comes back to knowing those word roots. And note that we're counting from the carbon that's closest to that carbonyl carbon. And we typically draw that at the top of our structure. There are different names for the different monosaccharides that we have. And so you can see that you can have those OH groups kind of sticking out on either side. We can have what we call epimers, which are two sugars that differ only in the configuration around one carbon atom. So an example of this would be our D-glucose and our D-mannose. You can see that they've got the OHs sticking the same ways, except for this one right here, where this has the OH on the right and this has the OH on the left. So we can refer to these as epimers. That is not a word that I care too much that you know. So, I mean, it's a good word to know, but it's not really that crucial. So don't worry about it. But do worry about kind of the number of the carbons that things have, because that is really just going back to knowing your word roots and be able to distinguish between an aldose. So in aldose, we have the aldehyde, and a ketose, where we have the ketone. Note that these are all D monosaccharides. So what do we mean by D? That's referring to stereochemistry. And so our bodies use the D enantiomers of monosaccharides. And so when we talk about an enantiomer, remember that's a non-superimposable mirror image. And so an example here is going to be our D-glyceraldehyde and our L-glyceraldehyde. shown with like these projections to show you where the bonds are going. You have the things that are up to the sides are coming out at you, and the things that are up and down, the vertical lines are going back. So you can see that these are non-superimposable mirror images. And specifically diglyceraldehyde and L-glyceraldehyde. So this is our simplest sugar. All of our other sugars are going to be named D or L based on whether they look like the D or the L-glyceraldehyde in terms of this kind of OH group, like right here at the end before you have your CH2OH group. And so this is going to be determining the D or the L. Our bodies use the D. Note that this is different than amino acids where our bodies use the L form. So we use L amino acids and D sugars. In fact, you might hear the term dextrose for glucose. And so D-glucose is sometimes referred to as dextrose, which I think doctors do just to try to confuse us. Do not confuse this DNL with levorotatory and dextrorotatory, similarly how we didn't want to confuse it with our amino acids. So amino acids and monosaccharides, they're sugars. They're both based on the nomenclature, based on the stereochemistry of glyceraldehyde, and they are not referring to the light bending. And so some D sugars really are dextrorotatory, so they rotate that light to the right, but others are going to be levorotatory, so they rotate the light to the left. I know, just to confuse us, right? But actually, it's easier because we can just simply compare the structures rather than having to have experimental data. But anyway, that would be D and or L. In a Fisher projection, so one of these straight lines where you have the things pointing to the left and right and up and down, then you're going to have it on the right to be a D sugar or on the left to be an L sugar. We're basically just going to be dealing with the D sugars in this class. So those are what we call Fisher projections. And so the lines that are going to the left and to the right are projecting out at you. So those would be our wedges. And then these lines going up and down, those are going away from you. So you can think about this is doing a back bend, and these groups are coming out at you. Again, you can see that we're numbering from the top to the bottom. This is an example of an aldose, and so you can see that we have the CHO. That's a common abbreviation for an aldehyde. This is the Fisher projection, but sugars often form these rings, and they cyclize through what we call hemiacetal or hemiketal linkages. So again, this is not a term that I need you to know, but it's a term you might see. The way that the linkages occur is that one of those hydroxyl groups in the chain is going to attack that carbonyl carbon. We call this carbonyl carbon that gets attacked the anomeric carbon. The O can attack from the top or from the bottom, and that will dictate whether we have an alpha or a beta version. And so if that OH group ends up sticking down when we have one of these, what we call a Hawthorne projection, where we have our oxygen in the upper right corner, this group, if this oxygen is facing down, this OH group is facing down, we call this alpha, and if it's facing up, we call it beta. These are both hemiacetal groups, and this is our anomeric carbon, that carbon that was the carbonyl carbon and that got attacked. Then depending on how it got attacked, you get your alpha or you get your beta. A very similar thing would happen if we had a ketose, but we would have a longer chain here instead of a hydrogen. Or it would be sticking down in the case of our beta. But the OH is what we want to look for when determining alpha or beta. And in the case of our ketoses, then we form a hemiketal and a ketal. When we form a glycosidic bond, we form the ketal. So again, this is just some terminology that you might see. Another terminology that you might see is going to be a pyranose. And so a pyranose is where we have a six-sided ring, whereas if we had a five-membered ring, that would be a furanose. And so we'll often see glucose in this pyranose form and fructose in the furanose form. Note that when we converted from our Fisher projection to our Haworth perspective, now we have that things that were on the right of our Fisher projection are facing down in our Haworth projection, and things that were to the left are facing up. So I think it's good practice to kind of go between trying to convert Fisher to Haworth perspectives, but I don't think that it's worth spending too much time worrying about it because that's just not what I want your focus to be. So I did want to introduce you to you though, because I think that knowing at least the alpha and the beta are going to be helpful when we talk about how we link these up. Because whether they're alpha or beta is going to help dictate the shape of the more complex sugars that form. But if we look at glucose, let's see how we can classify it. And so this is going to be like one of those Fisher projections, whereas these are going to be our Haworth projections. We count the carbons, 1, 2, 3, 4, 5, 6. We have 6 carbons, so we can call it a hexose. We look to see if it has an aldehyde or a ketone. We have an aldehyde, and so we have an aldose. And then we see when it ringifies, so we have this OH here that is going to attack our carbonyl carbon. And this carbonyl carbon that we have here, this is our anomeric carbon. And when we attack that anomeric carbon, now this was the carbon that got attacked. And this up here we have sticking up is this end group here. We have here the OH group is sticking up, so this is beta, and this sticking down here, so this is alpha. And both of these are going to be D sugars that had this OH group on the right. Okay, I think that's the bulk of the terminology that we need to get out of the way, so now we can move on to the more fun things. Because sugars often a lot of ways you can do fun things because of all those hydroxyl groups, all those OH groups. And remember that oxygen is a really great nucleophile. So we can add a variety of different functional groups. And so it's important to go review those functional groups. And so you can see here that we have amination or amidation because this is an amide bond. And then we have acetylation. This is one of our acetyl groups. And so this is called an acetyl D-glucosamines. This is an example of an amino sugar. We can get sugar acids where we have a hydroxyl group that gets oxidized, forms more bonds to oxygen. And here we have what we call a glucuronate. And so if you had a proteinated, you'd see glucuronic acid, which you might see in some supplements. And so this is an example of a sugar acid. We can also have reduction, so we're losing bonds to oxygen. Here, in this case, we would have 2-deoxy-D-glucose. So 1, 2, at the 2 position, we're losing an oxygen, so we're deoxy. This is 2-deoxy-D-glucose. And so D, again, is coming from up here. We could also classify this as being beta because we have this OH group facing up. So those were just a couple examples. And so we actually have a lot of different monosaccharides that we can have. Our body has 9 main ones. And these are the building blocks of most of the complex sugars that we have. And there's this kind of nomenclature for them. In addition to having various abbreviations, we have shapes, colored shapes. All of these can be made from glucose, and glucose is going to be represented by this blue circle. So the circles are going to be representing hexoses. We have N-acetyl hexosamines are going to be squares. Acidic sugars get diamonds, deoxyhexoses get triangles, and pentoses get stars. The shapes are going to tell you things about the lengths and the bonifications, and then the actual colors are going to be specific for specific monosaccharides. And so you'll see when you have like chains of monosaccharides, you'll see that they're often going to be shown in this simplified notation where you have these different shapes. And of course, it's only simplified if you know what the shapes represent, but thankfully there's typically keys. So we're going to focus now on just that glucose. And so remember, all of this could be made from glucose. And so where do we get glucose? So we can get it from a variety of different places. We can get it through our diet, from the interconversion of other monosaccharides. So we can make other monosaccharides from them, and we can make glucose from those monosaccharides. We can also make glucose from non-monosaccharides, so non-carb sources. We can call that gluconeogenesis. And then we can get glucose from breaking down our storage form of glucose. Our storage form of glucose is called glycogen. And when we break that down, we call that glycogenolysis, and that's going to give us some glucose that we can actually go and use in our bodies. And we're going to talk a lot about glucose and glucose regulation, and it's going to get complicated, but don't worry, we're not going to get there until we start talking about metabolism. So don't sweat it yet, but you're going to see how glucose can be used for a variety of different things. And even though it gets complicated, it's really cool, and we'll take it step by step, and hopefully it won't seem overwhelming. But you can see that when glucose goes into our body, or we make glucose in our body, we can link it up to form glycogen for storage. We can take it through glycolysis if we want to break it down for energy. A lot of people focus on just thinking about glycolysis, but glycolysis is happening as only a single option for glucose. And so we will talk about glycolysis, but we won't just talk about glycolysis. We'll talk about how we can link up that glucose to form glycogen for storage, so our glycogenesis, how we can take that glucose through what we call the pentose phosphate pathway, which is good for if you want to generate NADPH, which is a reducing agent that can combat oxidative stress. And it also provides things like nucleotides and DNA and RNA and coenzymes. It provides the precursors for those. Here we can see this hexosamine pathway. So if we take glucose partway through glycolysis and then take a branch it off, we can get those modified monosaccharides, and then we can attach those modified monosaccharides to proteins and lipids and get all sorts of really cool things. So lots of options for glucose. And now let's talk about kind of the simplest thing that we could do is just stick two of them together, right? It is this OH group that can, this OH group is what we call a reducing end. And so it can, the ring can open up here, the ring can get oxidized, and by getting oxidized, it's reducing something else. And so we call it a reducing end. Sometimes sugars will use this OH to form bonds, and sometimes they'll use another OH. Here, in this case of maltose, you see that we have this OH in the hemiacetal group, this one position, that's getting linked to this OH in the 4 position of this other glucose monomer. And so what we have here is we have a 1,4 linkage, and this one is not using its anomeric carbon. It's not using that hemiacetal group. And so it has a free hemiacetal group that can serve as a reducing end when these join together. And the way that they're going to join together is by a condensation reaction involving these 2 hydroxyl groups. You come together, you cook out water, you get this ether bond that we call a glycosidic bond. So just know that sugars are typically going to attach to things through those hydroxyl groups. Monosaccharides can link together to form disaccharides, and they can link together even more to form longer polysaccharides, and they can link to other things. And a lot of times that linkage is going to be through what we call glycosidic bonds. Those glycosidic bonds are going to be involving one of the alcohol groups, one of the hydroxyl groups on a sugar, bonding to something else through one of these glycosidic bonds, these ether bonds. So in this case, we have this disaccharide maltose, and we have this reducing end here. Sometimes, however, sugars link up in a way that does not leave them with a reducing end. So in the case of sucrose, you have both of them are using their hemiacetal group, their anomeric carbon, to form this linkage. And so you are not going to have any reducing ends, whereas with lactose and maltose, you do. Why am I saying this? Well, it's not so much that I care that you know what's a reducing end and a non-reducing end, but that terminology is going to be used when we talk about glycogen, when we talk about various polysaccharides, because the non-reducing ends are actually where we're going to be able to chew off from when we talk about breaking down glycogen, which is our storage polysaccharide. We want lots of those non-reducing ends so that we have lots of places at which we can go and chew off some sugar when we want some energy. So keep that in mind. Don't get bogged down. Just recognize that that is a term that you're going to see is reducing end and non-reducing end. And a non-reducing end has used both of those anomeric carbons, so it can't open up the ring, and it's going to be a place that we're going to chew from. That was if we had 2 monosaccharides, but we can hook up more. And so remember our word root oligo, which was few. So oligosaccharides have shortish chains of monosaccharides. Some examples of this are raffinose, which is the carb in broccoli that we can't digest, but bacteria in our guts can, and then they let off gas as a byproduct. Let's get more complex now. And so if we look up a lot of sugars, we get a polysaccharide. And so mono single, di 2, oligo, we've got a few. Now poly, we've got a lot. And so we have these long chains of sugars that have various amounts of branching. And the amount of branching is going to dictate the properties of that polysaccharide. And so often we call these polysaccharides complex carbohydrates, and we classify them as either structural or storage. And so storage for like energy storage. So what do you want with these? So with the structure, you want the strands to really stick together. Whereas with your storage, you want the strands to be easy to break down. So in our structural, we have these straight chains, and these straight chains are going to allow for more points of contact between the strands. And a lot of the contacts are going to be through hydrogen bonding that are helping kind of glue the strands together. Plants use cellulose in order to certify their cell walls. And then crustaceans use chitin or chitin, I can never know how to say it, but to stiffen their shells. And you can see that these both have these straight chains, But with our chitin or chitin, you have this basically modified one where you have this N-acetylglucosamine, which is going to have this group here that is going to offer up additional opportunities. So we've got this amide here. We have basically additional ways in which we can form interactions between the different strands of the sugar. But both of these are kind of going to have these long straight chains that are being held together. That's great if you want things sturdy, but then you only have those kind of single ends at which you could. Whereas if you want energy, you want lots of points at which you can start to chew from. And so you want to have some branching. And so in our bodies, we have a highly branched sugar called glycogen, and we'll talk more about glycogen in a minute. In plants, they have starch, and both of those are going to have these branching points, and they differ kind of in the amount of branching that they have. In our starch, we have a combination of two sugars, so amylose and amylopectin. You have these long chains, and you have these alpha 1,6 branch points. And so then with our glycogen, you have alpha 1,6, and then you also have alpha 1,4 linkages. And so you've got a lot of branching with the glycogen. So all that branching is great because now you have a lot of points at which you can go and chew off from. But why don't we just store glucose as is? Like, why do we have to hook it together anyway? One answer for you is osmosis. So think if we stored all those glucose units as individual glucose units, as individual monosaccharides, we would have a lot of little molecules inside of our cells. And what would happen is that because of osmosis, the water is going to come into the cells and that is going to kind of dilute out the glucose and it's going to make your cells swell and burst, and that would be bad. So we do not want to store glucose as individual units. We want to hook it up. The way that glycogen is actually hooked up is really cool. And we'll talk more about glycogen synthesis. And so we call that glycogenesis, and glycogen breakdown, that is glycogenolysis. Yes, there's a lot of words that have G's in them when we talk about sugar and metabolism. But when we have this glycogenesis, the making of glycogen, what it actually involves is making those sugar chains on a protein called glycogenin. So glycogenin is actually this protein at the center of this huge big network of these glycogen sugars. And so you've got all these branching sugars that are attached to this central protein, and it's really, really cool, and it's complicated. But there's some key enzymes involved with it that we'll talk about when we talk about glycogen metabolism. And if these key enzymes are disrupted, you can get various diseases. And so you might have heard about some of these glycogen storage diseases, Pompe disease, McArdle's disease, various types of metabolic syndromes that are caused by different mutations that are affecting different enzymes in the making and breaking of glycogen. So we'll talk more about that when we talk about glycogen and when we talk about metabolism in general, and we'll see how there's a lot of regulation that goes on when it comes to making and breaking that glycogen. So glycogenesis, the making of glycogen, glycogenolysis, the breakdown of glycogen. That glycogen is really good for quick energy because it has all of those different branch points. It's stored in your liver and in your muscles, but your muscles are going to break it down for itself for energy, whereas your liver breaks it down to deliver to the rest of your body. Glycogenolysis is actually only going to take you to this glucose 1 phosphate, which you can convert to glucose 6 phosphate and take through glycolysis. But if you actually want to ship out glucose, you got to remove that phosphate group, which is trapping it in the cell. And the liver is able to do this, but the muscles can't. So the muscles get to keep it and the liver ships it out. And again, we'll talk much more about this when we talk about glycogen metabolism. Another polysaccharide you might see is agar and agarose. And so agar is often used in the lab and it's often used as a food thickener. And it's a mix of two sugars, agarose and agaropectin, two of these like complex polysaccharides. Agarose is long, mostly consistent and neutral, which makes it really, really great for making agarose gels to separate DNA by size. DNA is negatively charged, and so you can send it through a neutral gel mesh in order to separate the DNA fragments based on their size. Agar, on the other hand, is going to include agarose, but also agaropectin. And agaropectin has a lot of those modified monosaccharides. It's got groups like sulfur groups as well as methyl groups. Those sulfur groups are negatively charged. Now imagine you try to run your gel electrophoresis, your DNA is going to get repelled by that, and the negative charges of your gel might actually make the gel start moving towards the positive electrode. So not good for your agarose gels, or not good, I guess, then for your agar gels. Instead, we want to use that agar more for places where we want to have, or we at least don't care if we have those groups. And so we make agar plates like petri dishes for bacteria. Those plates are really good. Those modification groups, like it's great. They soak up a bunch of water. You get this nice gel. The bacteria are happy, unless you put antibiotics that kill them. But we're able to make this nice gel, and we don't have to worry about having this actual agarose. We're good with this agar. We don't need to go purify out the agarose component. But you don't want to use the agar for a gel electrophoresis. So that was when we just had sugar. So we've got sugar, we got some modifications on the sugar. We have different lengths of sugars, but we're just dealing with sugars. Now let's get to glycoconjugates. So when we talk about conjugates, that's when we have something covalently bound. And so in a glycoconjugate, we have a sugar covalently bound to something. Often these linkages are going to be linked through a nitrogen group or an oxygen group. And so we can think about glycoproteins, where we have proteins that have glycans or sugar chains attached to them. Often, rather than just simple chains, you have these kind of branch structures that are attached to the N in an asparagine, or they're attached to the oxygen in a serine or a threonine, or a hydroxylated lysine or proline. And you get these linkages, these glycosidic bonds between the sugar, the glyc part, and then whatever else. And so in this case, it would be the amino acid. This allows you to link these trees of sugar onto proteins. And so typically what happens if there's a protein that is going to be secreted or membrane bound, they're often going to have glycosylation, and they get this kind of tree of sugars attached to them. And the same precursor bonsai tree gets attached, and then it kind of gets whittled down. It can get whittled down in different ways to give you different properties. But if it doesn't get whittled down correctly, then what happens is it triggers it to get degraded. Your cells have what's called the ubiquitin proteasome system, where this little protein called ubiquitin gets attached to proteins. And so you get chains of proteins on your protein, and then it gets degraded. And that was happening in this case, in some cases, because that tree didn't get whittled down. And you can see that they're using that shorthand nomenclature, those symbols, to represent the different triggers. Proteins on the surface of viruses are often heavily glycosylated, and that is going to shield them from the immune system because they kind of hide the protein part that the immune system typically recognizes. One more terminology note is that glycosylation refers to the enzymatic addition of sugar chains, whereas glycation refers to the addition without enzymatic help. And so like HbA1c, which is hemoglobin with glycation on it, that glycation happens because there's a lot of glucose in the blood. And so in people with diabetes, especially, they have high levels of glucose in their blood. And that glucose can then non-enzymatically get linked to hemoglobin, and that's going to give you this glycation that can be diagnostic of diabetes, because if you have a lot of glycation, that indicates that you've had a lot of glucose in your bloodstream, and that glycation actually sticks around and serves as evidence that you have had blood sugar levels that were too high in the past. And so it's not just an instantaneous measure, but a measure of sort of the amounts of glucose in the bloodstream for the past 2 to 3 months. So that's an example of a glycoprotein. We had mostly protein, and then we also had this kind of sugar tree attached. Let's now move on to talking about where the sugar gets to play more of a starring role. How about proteoglycans? So proteoglycans is where you have that long chain of a protein with lots of sugar chains attached. Some examples are heparan sulfate, chondroitin sulfate, and keratin sulfate, as well as mucus. The sugars are going to play the starring role. And so glycosaminoglycans are large polysaccharides with a lot of modifications. Those can be amino sugars, oxidation to carboxylate groups, addition of sulfate groups. And the different kind of codes of these modifications provide binding sites to proteins in the extracellular matrix. And so these are really great for acting as scaffolds. And the incorporation of sulfate groups allows them to soak up water and form useful gels. So we don't have agar gels in our body, but we do have gels in our body. And those are often going to be made up of things like our chondroitin sulfate. Those things are going to form those nice gels that lubricate our joints and act as shock absorbers. So let's look at peptidoglycans. So peptidoglycans are where you have these little peptides that are linking together chains of sugars. In bacterial cell walls, they have this peptidoglycan layer in this kind of space in between their membranes. They have these modified monosaccharides that are linked together to give us these polysaccharide chains that are held together through these shorter chains of peptides. And so remember, peptide is just a word we use for a short strand of amino acids. And so here we have short strands of amino acid linking together long chains of sugar. And that would be an example of a peptidoglycan. Speaking of which, connecting this back to our talk about enzymes and enzyme inhibitors, beta-lactam antibiotics like ampicillin and penicillin, they actually go and they inhibit the transpeptidase enzyme that is responsible for making the linkages between some of these peptides. And so if you inhibit that, the bacteria can't make those sturdy cell walls, and then they die. We don't have peptidoglycan in our cell walls, but we do have these phospholipids. And so lipids are able to be conjugated or covalently attached to sugars as well. So we'll talk more about lipid structures, but they have a variety of different groups that offer opportunities for linking to proteins. In the case of bacteria, they have these lipopolysaccharides that are referred to as LPS or endotoxin. And you have these saccharides, these sugars joined to the membrane, phospholipids that are going to shield the gram-negative bacteria and make our immune system react to it. That's bad. But we also have some glycans in our cells that are helpful. We'll talk more about lipid structure, but basically they have these long hydrophobic tails and these hydrophilic heads. And this makes it so that they make this sort of sandwich shape when you put them in water, with the hydrophobic parts getting excluded from the water and therefore pushed together to free up the water molecules. Ta-da! Hydrophobic effect. But going back to this, they also have that hydrophilic head. The hydrophilic head, water's going to want to hang out with, it's going to be on the surface. And the hydrophilic head is going to offer opportunities for bonding to glycans. And so you'll see this like GPI linkage, glycosyl phosphatidylinositol. So basically, you have one of those head groups can link up to a sugar, and that can also link up through a protein. And there are various other sugar modifications on lipids, and we'll not go into those. So those were the examples of glycolipids. We have different types of glycoconjugates. We have those kind of three types that are associated with a sugar and a protein or a peptide. Our glycoproteins have the protein as the main component and then the glycan chain attached. Our proteoglycans have the sugar as the star, and you've got lots of chains of sugar attached to a long strand of protein. These are great for things like making gels that make your joints not hurt and that absorb shock. And they can serve as a sort of code based on the amount of modifications and the presence of various modifications. They can make a sort of code in your extracellular matrix, so the area outside of your cells. Peptidoglycans are here where you've got chains of these sugars that are joined together through these short little peptides. An example of that was the peptidoglycan in bacterial cell walls. And glycolipids, where we have sugars attached to lipids. So there are lots of different options for linking sugars to things, and sometimes the same sugar is linked onto different things. This is involved with ABO blood groups, and so the ABO gene is located on chromosome 9, and that's not important to know, the chromosome, but it's important to know that there are different versions of this gene, and that this gene encodes for a glycosyltransferase, so a sugar adder. This enzyme is going to be responsible for adding sugars to a core sugar chain. And so it's you have this chain, and then this ABO gene, it codes for a glycosyltransferase enzyme that adds an additional sugar. The A version is going to add N-acetyl-D-galactosamine. The B version is going to add a different sugar, galactose, and the O version is a dud. It doesn't add anything. And so you only have that shorter chain you start with. And depending on which of these sugars that you make, therefore, your body is going to get used to seeing different sugars on your cells. And the sugars are going to be able to, however, serve as antigens, so things that antibodies recognize and bind to. And so if you have a sugar introduced to you through, say, a blood transfusion that is not one that your body normally makes, then your immune system is going to bind it and it'll cause problems. And this is the basis for tests to see what blood type you are before they actually give you a blood transfusion so that your body doesn't react. So yes, glycobiology is sweet. There's some not so sweet things like all the terminology, but that terminology is just so that you can understand the way that things are displayed so that you don't get bogged down in that when you're trying to understand all the cool stuff. And so the really fundamental things to take away are that monosaccharides are individual sugar units, and the most fundamental one is D-glucose, which is often referred to as blood sugar. These monosaccharides can be modified via amination, carboxylation, amidation, acetylation, all those good functional group additions that offer up additional kind of opportunities for fun things to happen, additional places for things to bind, and can even serve as a sort of code. Monosaccharides can be joined together through glycosidic bonds to form disaccharides, oligosaccharides, and polysaccharides. Depending on where the linkages occur and with what stereochemistry, they'll provide different shapes instead of different functions. Polysaccharides can play storage and structural roles, and the main storage carbohydrates are glycogen in animals and starch in plants. They have high amounts of branching with lots of ends to chew from for energy. In contrast with structural carbohydrates, you don't want the branching that would be interrupting the bonding between the chains. Instead, you want long, straight chains. And examples of these are cellulose in plants and chitin in arthropods. They have a highly ordered structure with straight chains held tightly together through hydrogen bonds. Saccharides can be covalently attached, so we call that conjugated, remember, to peptides, proteins, and lipids to form glycoconjugates. These are named based on their major and minor components, with the names starting with the minor one to modify the major one. And there you go. Sugar science. Sweet!